Hi, I'm Feiran. I'm a PhD student from Sisbel Chalmers University of Technology. And this video is for a popular science project in Chalmers to introduce to public about our research. And I'm glad to introduce to you my research, which is the genome scale metabolic model of yeast. Metabolic model is all about metabolism. And what is a metabolism? For human, we eat fats, carbohydrates every day, and to generate the ATP, and which is energy to sustain our daily life. And in that case, the metabolism for human cell is a conversion from the nutrients to the ATP and the chemical waste, such as the carbon dioxide and the water. I'm interested in yeast. Yeast is one of the most important microorganisms which can be used for wine production. And the metabolism for yeast is to convert sugars to carbon dioxide and alcohol to generate ATP and the cell biomass. The process of metabolism contains thousands of reactions and millions of molecules. And in order to study that in a systematic way, we generate the genome scale metabolic model by converting all these equations, all these reactions into a mathematic matrix so that the computers can read and simulate. Gene reaction metabolites are three main elements in the gene. The reaction and the metabolites are connected to each other by a S matrix, as we just showed. And the gene and the reaction are connected by the GR rows, which indicates the genes associated for a certain reaction. Then we group reactions into different subsystems by their functions. And we can simulate flux, which is the rate for certain reaction. Gene can transcribe to RNA and can translate to protein. And so far, I have shown all the elements in the gene. And through those elements, we can actually map all this data generated in systems biology to a single gene. In that case, GEM is a wonderful platform for data integration and data analysis in systems biology. GEM can be applied to get the metabolic engineering. For example, we can identify which genes are important for the wine production. And we can also use the GEM for the enzyme function identification. We can use GEM for evolution analysis to tell the differences and the divergence from the yeast for wine production and yeast for milk production. And we can predict the biological phenotypes through the GEM. So for example, we can tell the which reactions and which pathways are active during the alcohol fermentation. Ever since the publishment of the first GEM in 90s, the development of GEM has been split in two directions. The first is in the species number. So we apply the GEM method to more and more species, from bacteria to fungi, yeast, and even to the human cells. On the other hand, the GEM has developed to include more and more processes besides the metabolism, as we just showed. And right now, we even have the whole cell models for several species, which contains almost every process in the cell. And my research also divides in two directions. The first one is to expand the model for baking yeast to other different strains of the baking yeast and to different yeast and fungi species. For example, the milk yeast and the wine yeast, and also the other yeast. And the other directions to include more processes and constraints. So I include the enzymatic constraints for the model for baking yeast and also include the enzymatic constraints for the different yeast and fungi species models. After that, I will introduce to you the most complex model that I have constructed in my PhD study, PC-sec yeast, which contains a lot of processes that we will discuss later. So let's start with the first and simplest one, yeast 8, which is a model, which is a normal gene for baking yeast. Actually, we are not the first one who decided to generate a model for baking yeast. There are already published like more than 10 models for baking yeast. And why we want to generate a new variant. That is due to during these years, the knowledge for baking yeast is growing rapidly. And we want to integrate all this newly acquired knowledge also in the model. So we designed this new pipeline so that we can update the yeast 7 to the current variant yeast 8 by adding new reactions and filling more gaps. After that, I decided to expand the model for baking yeast to other strains in the baking yeast and other species in the fungi and yeast species. And the method is basically the same, except we do more refinement and curation in the yeast and species model reconstruction, since compared with these different baking yeast strains, those yeast and the fungi species are more phylogenetically distant. After reconstruction of those models, we are able to tell the differences among the baking yeast strains from different origins. We can use the different yeast species models for the evolution analysis. Then so far I have tell all the development in the direction of species. Then I will start to talk about the development in another direction, which is process coverage and constraints coverage. So let's start talking with what is the constraints. If we don't constrain the model, it means that the model will have a unconstrained solution space. It means that every reaction can operate in any specific rate the model wants. And if we want to let the model have a solution, actually close to the real solution, then we need to add the certain constraints. And the normal gene have these constraints, so we will have a allowable solution space, which is much smaller content with compared with the unconstrained solution space. Then by optimization, then we can have the optimal solution. Then we are hoping the optimal solution would be close to the experimental solution. But actually, the allowable solution space is also is still large. So we would like to include more process and more constraints to further shrink the solution space. And here I will start to talk about the first new constraints that we will add to the normal gene, enzymatic constraints. We will generate the enzymatic model for the baking yeast based on the normal gene of baking yeast. And we will generate the enzymatic models for 300 yeast and fungi species based on the normal gene of 343 yeast and fungi species normal genes. And let's start talk about what is the enzymatic constraints. Every reaction is every metabolic reaction is catalyzed by metabolic enzymes. And if we don't add this enzymatic constraints, which means the reaction can operate in unlimited amount, but it's not the case because we know the enzyme has the maximum capacity or maximum rate, which is the keycat number here. So after we adding the maximum rate constraint in the model, then we see that the solution space is shrinked and the predicted solution is close to the experimental solution and the error is reduced. The method seems quite simple if we are talking about one species, But if we want to apply this method to multiple species, for example, here 300 different yeast species, then there came up with some issue. And if we look at these constraints, then we know that it is highly dependent on the KiCad parameter, which is the maximum rate for one certain reaction. So if we look at how many numbers do we have for the KiCad number for one single species, then the 300 species are actually not in the top list. So in that case, which means the KiCad parameters are not sufficient to generate the enzymatic constraints for all these yeast species models. So in that case, we generate a deep learning method to predict the KiCad parameters for all these 300 yeast species. And after that, we are able to generate the enzymatic constraints for all these yeast species and generate the enzymatic model. And after that, we are able to identify the enzymes or proteins which are important for a certain phenotype. Here I showed this CIT and PYK, these two enzymes are very important or directly linked to the Crabtree effect phenotype. Actually, the Crabtree effect phenotype is the main phenotype which contributes to the alcohol fermentation. And at last, I would like to talk about the most complex model that I have constructed. So besides the metabolism I just show, and also the enzymatic constraints, and I did more processes in this model, including the protein translation, protein modification, and protein salting to the final destination of this protein. And by doing so, we have a much more complex model so that we can simulate the more delicate phenotypes of yeast. And here I show, using this model, we are able to identify some gene targets to improve the alpha amylase yield in yeast. Alpha amylase is an enzyme and commercial enzyme, so that it's sometimes it's commercially produced in yeast, bacteria, and fungi. Here we show that by manipulate those genes, then actually alpha amylase can be improved. A little summary. So Genome is a wonderful platform for systems biology, and Genome can develop in two directions. The first one is to expand to more species. The other one is to expand to more process or more constraints. And the third one is if we met some uncertainties in the model constraints, then we can always try to find a way to overcome that. For example, here I showed the KiCad, or the maximum rate for a reaction or enzyme can be overcome by deep learning estimation. And finally, I want to express my thanks to Jens, my supervisor, Edward, my co-supervisor, and Hongzhong Le, Yu Qi, Yan, Benjamin, Jamie, Ivan, Gang, Ibrahim, for their collaboration in those wonderful projects. And I would like to thanks to Amir, Rasul, Hal, Pinar, and Michael for their support and discussions during the work. And I would like to express my thanks to all CISPL members for this wonderful family. And at last, I would like to thank DDD-CAF Foundation and Novo Nordisk Foundation for their support. And that's it.